Hello everyone and welcome to today's webinar titled RNA Bead-Based Assays to Detect Tumor Gene Amplifications in Blood-Derived Exosomes. This webinar is part of the Cell Bio 2021 and I'm Susie Valdez of LabRoots and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by Luminex. For more information about Luminex, go to www.luminexcorp.com. So let's get started. Before we begin, I want to remind everyone that this event is interactive and we encourage you to participate by submitting as many questions as you'd like during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. And if you have any trouble seeing or hearing this presentation, simply click on the Help Desk button located at the bottom of your screen within the navigation bar or from the lobby. As a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education's credit link located at the Abstract window below the presentation window and follow the process to obtain those credits. Without further ado, I'd like to present our speaker today, Dr. Godfrey Grech, an associate professor with the Department of Pathology, Faculty of Medicine and Surgery at the University of Malta. For a complete biography of our speakers, please visit the Biography tab at the top of your screen. Dr. Grech, welcome. You may now begin your presentation.
Thank you very much for the invitation to present at the Cell Biology 2021 event. Today I will be taking you through the path from basic research in cell biology, and where we actually measure and study specific mechanisms of disease in oncology, and moving towards actually detection of these characteristics in blood using blood-derived exosomes, extracellular vesicles that are being shed by cells. So this is the path that we actually take. Of course, we start off with basic research where we study the cellular biology and the mechanism of disease. And once we have identified specific biomarkers that will give us information that can predict disease or on therapy outcome, or actually biomarkers that can be used to monitor patients, then we trigger immediately the path towards validating these biomarkers using patient material, patient data, and with all this information we will translate into a pipeline, a workflow, in which we can actually then measure these biomarkers depending on the specific question. So ultimately we will validate these diagnostic or theranostic biomarkers towards eventually commercialization. Now, our group have been studying the particular phosphatase, the phosphatase protein 2A, PP2A. PP2A is a complex, a phosphatase complex made up of three subunits. You have the A structural subunit, then you have the B, which is a regulatory subunit, and the C subunit, which is the catalytic subunit. And this complex is directed towards specific targets. In this case, I'm showing that the targets are actually the PI3K ACT mTOR pathway, and they dephosphorylate these targets. Now, when the complex is inhibited, this particular mechanism, this particular signal transduction is sustained and promotes survival, proliferation, and also differentiation block. Now, previous studies we showed that specific proteins, these are actually regulators, so it's one of these subunits, but in this case these are inhibitory regulators of this particular complex. And our journey started with actually identifying the role of alpha-4, or also known as IgBP1, in shifting the phosphatase activity towards different targets, but away from the mTOR pathway, resulting in a differentiation block that was shown on an erythroid progenitor model. Today we know a lot about these inhibitory subunits. Alpha-4 is one of them. SET has been shown to be extremely important in blast crisis in chronic myeloid leukemia. And CYP2A has gained a lot of interest in solid tumors. And this is where we are currently working with CYP2A by overexpressing it in various cell lines and studying the feedback mechanisms in various cellular models. In fact, we have now at hand cellular models which are breast cancer cell lines in which we constitutively expressed CYP2A, and we looked into the phosphoprofiling due to the effect of this overexpression. So the importance of PP2A in breast cancer led us to looking at the sensitivity of cell lines. In this case, I'm showing the triple negative breast cancer cell lines and their sensitivity to PP2A activator FTY720. Now, as you can see in these graphs, so the graph A shows TNBC cell lines that are sensitive to FTY720. There is the percent cell viability on the y-axis, and on the x-axis we find the concentration of FTY720. And you can actually appreciate that these three cell lines in figure A show that you can actually reach the IC50 at very low dosages of FTY720, two of which even less than 1 micromolar, whilst the TNBC cell lines in figure B show that they are not sensitive to FTY720. With one of the cell lines showing that there is a change, or there is an IC50 reached at 10 micromolar FTY720. So basically here we can see that there are TNBC cell lines that are sensitive and those that are non-sensitive, and actually we studied further these 6 cell lines as 2 groups. So we can see that we have the NDAMB231 reaches an IC50 at 1 micromolar, BT20 at very low dosages of 0.1 micromolar, and HS570 at 5 micromolar. And we compared these 2 cell lines using transcriptomic datasets. So we did RNA sequencing experiments to look at differential gene expressions between these 2 groups. And from these transcriptomic analysis, we identified a list of potential markers that are associated with sensitive cell lines to FTY720. Our understanding was, or our hypothesis was, that these markers are actually markers of low PP2A activity, and hence we continued with the experiments showing, and we actually showed that these sensitive cell lines have very low PP2A activity, which I will show in a minute, as compared to the non-sensitive. And then we utilize these differentially expressed genes into multiplex panels to compare these cell lines and even move on to patient material. So as part of the multiplex panel that we actually selected, we added the subunits of PP2A, and here we can see that we added the PPP2CA, which is the catalytic subunit, and the blue boxes. Then we have also regulatory subunits, the R2A, which is the light blue, and also the inhibitors SET binding protein 1, CYP2A, and AMP32A. And when we look at this data set, in which we are here, the figure shows normalized expression to housekeeping genes, and compare as a group of cell lines the non-sensitive to sensitive, the distribution of the PP2A complex and its regulators, we have a slight significance of CYP2A high in the sensitive group. But this data shows us that it is extremely difficult to compare the markers at group level, so the sensitive as a group and the non-sensitive as a group. I'm saying this because if you then look at specific cell lines, we can see that the 231 breast cancer cell line actually has a very high expression of CYP2A, while in the case of BT20 we see a very low level of the R2A subunit, meaning that different cell lines would have different deregulation parameters for the PP2A complex. Hence, this shows us that it's extremely important to look at the different cell lines, and also when we look at a group of cell lines and eventually patient material, we definitely would need to have a multiplex panel in which we have all the subunits that show the complex, but also addition of a panel of markers that show low PP2A activity, and in fact that is what we were after. And therefore we moved on to multiplexing, and we use this Quantigene plex assay from Thermo, which works on the XMAP technology using the Luminex platforms. So the Luminex XMAP bead-based multiplexing can go up to 80 genes. In this particular experiment we started with 40 genes. It is a nucleic acid sandwich hybridization method in which you have your target that you want to capture. Then you would have different capture beads, specific probes and capture extenders, which would specifically bind to one of the targets of interest. Now, once it is bound to this bead, then through label extenders, a DNA scaffold is used, in which, using a preamplifier and amplifiers, you will have this whole scaffold of label probes, which would eventually, through streptavid and PE, would eventually give us a signal. So basically here what we're doing is we are not amplifying the target as we do with PCR. Here we actually amplify the signal. And then using the Luminex platforms we measure the fluorescence. Now, why we want to go for multiplexing? It's basically because cell biology is dynamic, cell biology is complex. And when you look at some of the cell lines that we used, and here we divide them as primary cell lines and metastatic cell lines, those that are sensitive to FTY720 are marked with an asterisk here. Okay. And you can already see that this sensitivity is not based on the expression of epithelial markers, because here you see E-cadherin and EpCAM. This is the median fluorescent intensity that we get from the Luminex, so this is normalized to housekeeping genes. And here you can see that these are epithelial because you have expression of E-cadherin and EpCAM, whilst the others are not. So this is only showing 2 of the plex. Then we looked at, this is the same graph, but now I'm extending the scale here. And here you see, like, if we look at specific mesenchymal markers, like NCAT-hearing, SNI1, SNI2, FN1, and twist, you can see that now this shows up as a mesenchymal marker cell line, and that is sensitive to F2Y720. When we look at vimentin, which is another mesenchymal marker, you see that this is positive for vimentin. So this is always the same, but we are scaling it up to see the markers. So this shows the importance of actually having multiplexing, so that you actually have a good classification. So these are called classifiers of the different cell line models that we use. Using only one marker or two, that would not provide us with the full information. So coming back to our interest to show the low PP2A activity, what we did is we compared these groups at a transcriptomic level, and we identified differentially expressed genes between sensitive and non-sensitive. Here on the left you see a graph in which we actually pulled down the complex and measured the activity, the phosphatase activity, against a substrate in vitro. And as you can see, the sensitive have low PP2A activity and the non-sensitive have high, but this is something we cannot do on patient material. Of course, we can do it on cell lines, but not really on patient material, and therefore we wanted to find this marker that discriminates between these activities. Here we just add this as a positive control because we know that K562 have actually low phosphatase activity. So when we did all this transcriptomic and checked the networks and gene networks, we encountered this particular panel, and we put our interest in Aurora kinase A and KIF2C as the markers. When they are highly expressed, then we always have a low PP2A activities in our cell lines. And of course we extended studies with inhibitors and activators, and we looked at all the possible designs to show that our kind KIF2C are really excellent markers for low PP2A activity. And you can see it here. Okay, so these are non-sensitive and sensitive, and this is the activity, corresponding activity. So then once we actually found this marker, we said, okay, how important this marker is in the context of breast cancer patient tissue. And here where we started using the publicly available data sets, in this case the TCGA, and here we're looking at RNA sequencing normalized expression values, looking at matched normal to tumor. So we only looked at those samples in which they have matched normal and tumor, and we compared the expression of ARCA and KIF2C, ARCA in yellow and KIF2C in red. They seem to move always together, but the most important is that there is an increase in these markers when comparing normal to tumors. So that was already very interesting to see. More interesting is that you have a higher magnitude in luminal B, which is known to be the most aggressive, and high in Ki67 proliferation marker, and also in basal. So this is very interesting to see that we actually have markers that shows us a difference between tumor and matched normal and the different subtypes. Of course, we also did a lot of work on our TNBC patient material, and to our surprise, ARCA is expressed at a high level in more than 72% of TNBC patients. Here we used immunohistochemistry on FFPE whole sections. We did the H scoring, and we used 160 cases for this particular experiment. And here you can see the increase in the H scores of Aurora kinase A, or ARCA, in the TNBC patients. Now, we kept on looking at some important data sets as well, and here again we'll move on to the TCGA dataset. This is something that we've seen from our FFPE material, where we saw a correlation between the expression of AURCA and one of the inhibitors of PP2A in breast cancer patient, and this was a correlation between AURCA and CYP2A. And here we extended this to also TCGA datasets, where we show here in the left that AURCA messenger RNA expression correlates with the expression of CYP2A, which is quite significant. Now, it's a well-known fact as well that AURCA is amplified in various cancers. And in fact, the AURCA copy number variation correlates excellently with the TPX2 gene. That's because they are on the same chromosomal region and they are generally amplified together. I'm saying this because we did a lot of work on gene amplifications in various cancers. There's a high frequency of these amplifications, and today we know a lot about gene amplifications. So we started this project. What we did is, again, we wanted to go for multiplexing, so not really studying one, so we do a simultaneous analysis of various gene signatures. And during the experiment, we used even 40 plexes. We looked at these common amplified genes in CRC within an 18 multiplex assay. We looked at those that are involved in metastasis and invasiveness, and also therapeutic resistance. This is a specific cohort of CRCs, and this is patient cohort of CRCs, both primary tumors and matched biopsies from metastatic lesions. So it's really a very specific cohort, meaning that these have already had a metastasis. But we went back to the primary tumor, and also we actually had the metastatic lesion. Both in FFPE, so formula fixed, paraffin embedded. It's good to say that with the quantigene assay we can actually run FFPEs, because it's not you can actually read these gene amplifications and gene expressions in this case, and using even degraded material. So here we show that there's a huge amount of TPX2 represented in this particular group of patients. So TPX2 was overexpressed at an mRNA level, okay, which is for us reflects that there is a gene amplification. So overexpressed compared to normal colon. Then if you take TPX2, FZD1 and IGF2, we have all the cohort together. So that's a nice panel for these specific types of tumors. Then we looked at the metastatic lesions, where we find 50% of CRCs moving to the lung, showed a very high expression of TOP2A. CCND3, cyclin D3 was overexpressed in lung metastasis. Those that are exclusively found in liver metastasis, CDX2 and KIT in this case, and MYC was found consistently gained in metastasis over the primaries in all the types. And then, okay, so we had sort of an idea of the gene amplifications at primary and metastatic, and then I wanted to move on with other projects to really look at innovative molecular profiling assays, to look at distinct circulating tumor cells and also exosomes. We wanted to develop an actionable panel, multiplex panel, for these circulating cells or vesicles. So Okay, so I'm not going to take a lot of time here. So liquid biopsy is you have a solid tumor and these will shed exosomes. You have to have, there's also apoptotic bodies, DNA, RNA, okay, cell-free RNA, and CTCs. So these are all the different ways how we can measure it. We focused on CTCs, but in this particular presentation I will focus on exosomes. Now, in general, exosomes are used to read microRNAs. In our case, we used it to read messenger RNA expression to reflect gene amplifications which originate in the primary tumors. CTCs, as you know, can be pulled down with AbCam antibodies, although in our research we also showed that some of them are AbCam negative. So here again, putting multiple antibodies would be necessary depending on the patient. And first we wanted to look again into the amplifications in CRCs. So we have cell lines, we have blocks, so we have a whole workflow in which we can actually do IHCs, look at markers, at histochemistry. For instance, this is an SKBR, which is HER2 positive cytokeratin 19. And this why we wanted to really look how sensitive our technology is so that we move on to exosome research. For instance, if we use AbCam, we will never capture such cells like the HS5780 because it is AbCam negative. So we managed to detect amplifications as expected in cell lines. For instance, if we look at SW48, we know that there are the MET and the GFR amplifications. We see them at a cellular level in the cell line itself. That's normal. Okay, but then our interest was to actually see these reflected in the exosomes from which that are generated from these cells. So we started off with another plex. Here we have a 6-plex looking at CTCs. We have AbCam cytokeratin 19. We have FN1. This is a marker that we really work a lot on, and we've shown that this is involved in early invasiveness. So then we had HER2 there, ER, because we had breast cancer cell lines for this particular study, and MUC1, and we use Luminex technology to read these. Now, what's interesting here, we did a titration experiment with different cells. So here you see the MFI, mean fluorescence intensity, against cell number. Okay, and here we have blue is AbCam, orange is cytokeratin 19, yellow is FN1, and green is HER2. Now, what we can see here, if we look at SKBR, which is HER2 positive, we can see that the green one, HER2, okay, is very high, cytokeratin is very high, AbCam is high, and it is negative for FN1. So if we have FN1, we don't find these. If we use only AbCam, you can see that the magnitude is not as high as using HER2 and cytokeratin 19. So in this case, we of course promote the use of cytokeratin 19 and HER2 for this particular cell line, because we can actually detect down to 5 cells here with this quantigene assay. Whilst using this cell line, we can see that again AbCam is high, but you don't detect lower than 40 cells. Whilst with HER2 and cytokeratin 19, we can go down to 15 cells. That all depends on the expression level of these markers. Now look at this. This is totally different. HS5780 is negative for everything except FN1. So, and with FN1 we can even read 5 cells. So the importance of multiplexing in actually detecting these CTCs. And then, okay, we moved on to exosomes, and here is where one of our industrial partners, Omnigene, is working and helping us with, is to put together a workflow, automated workflow with microfluidic miniaturizations to increase the sensitivity, so that we actually read the exosomes and the plasma by immunoprecipitating them with specific beads. So the exosomes are these membrane-bound extracellular vesicles from endosomal origin. Here you can see electron microscopy of these exosomes. Keep in mind that exosomes are extremely small, so 30 to 120 nanometers. If you have something which is in the sort of 2 microns, those are apoptotic bodies. And in general, microvesicles are sort of indicating all these types, but we are working with exosomes. Now, exosomes are from endosomes, so they have specific markers. So mainly we worked with CD63 and CD9. Yeah, we have streptavidin donor beads. We biotinylate antibody which would bind to exosomes. In this case, it's CD9. Then we have, of course, the exosome capture complex. And from this exosome capture process, we are enriching the exosomes from plasma. Okay, and once they are captured, then we can also see these captured exosomes in flow cytometry, okay, using, for instance, an CD63, which is conjugated with a FITC. And this is the exosome detection complex that we see on flow cytometry. And it's easy to put together. Okay, you can look at CD63, FITC. You can also use other markers like HER2 if it is a HER2 positive cell. And this is what you see on a flow cytometry. You would have these single beads which are loaded with exosomes. You will see the double beads and the triple beads, but even less and less every time. So we can gate on these single beads, and we can see actually the positivity of these exosomes to CD63 when we spike these exosomes in blood. Okay, so here we can see a shift. This is the no exosome control against the exosome sample. So this is HS5780 produces exosomes. We know that because we measure them as well with nanocytes, but they are not CD63 positive. Okay, so again, the importance of multiplexing to actually avoid negative results. Okay, so then we moved on, like, okay, if we produce exosomes from these cell lines, what is the representation of these panels and markers? So if we look, if we run the quantigene assay, which is using the median fluorescence intensity, using EpCAM, cytokine 19, FN1, and HER2, SKBR, it's positive for all of them. But the exosomes, you can see cytokeratin 19 and HER2. Okay, so same trend, but at a smaller scale. Okay, but we still actually can detect them. Why is HS570, which is in this case for this particular panel only positive for FN1? You also see the same trend in the exosomes which are derived from this originator cell. And when you compare 40 different genes in a multiplex fashion, and here we have identified and created panels which we call CRC amp for amplifications and diagnosis, CRC invasion, which are important gene amplifications as a result involved in invasion, and CRC mets, which are in metastatic disease. So when we put them together, then we find those that are amplified. In this particular cell line, we have these which are amplified, and you can see that if you plot the MFIs from 250 nanograms of RNA, it has a very nice regression with MFIs resulting from exosomes. Now, of course, you find some outliers, but in general, for gene amplifications, you will actually have this nice correlation. So yes, we can actually measure the gene amplification in exosomes that originate from specific. Well, we didn't stop there, so we actually have set up a method a workflow to actually isolate them now from patients. In addition, we also can detect the amps in 0.5 millimeters of FFPE cores, and this we showed the result before, primary versus metastatic tissues, in which we found these different gene amplifications in metastasis. But of interest, we managed to detect these particular 4 gene amplification in some of our patient-derived exosomes using only 0.5 mils of plasma. And this is an effort between our university and Omnigene, in which we are setting up this multiplex assay to actually isolate these exosomes and read them from patient plasma in CRC. So what we are currently, what I've put together here is we're starting from biology. We understood that there is a specific phosphatase, and we can actually look at specific markers. And those markers we went into the interest of gene amplifications because it was involved in gene amplifications. And therefore we wanted to read cellular biology at a tumor level, but also in the tumor-derived vesicles and cells in blood. So we know that there are various clinical unmet needs like equivocal diagnosis, heterogeneity of tumors, looking at the dynamics, progression of disease and resistance to therapy. Okay, there are also problems to validate novel biomarkers, and therefore multiplexing and this high throughput methods can be used to actually do that from various sources. And of course, we're put together an enabling technology together with ThermoFisher and Luminex as enablers, and we go after high accuracy, precision. Most importantly, and very important, is that we are getting a digitalized result, okay, in which we actually can also use it for patient surveillance, because we can read less than 15 cells. We can read exosomes now, we know as well. Okay, we can measure markers in low quality material and low input from FFPE. So this is all done and tested. So for these studies, it was only possible through the efforts of various parties, including the industrial partners, the enablers, Luminex for the XMAP technology for the multiplexing, ThermoFisher for their quantigene assays, Omnigene for putting together workflows for the exosome research, then the whole research group, including Christian Sherry, Janice Sherry, Michelle Chechi, Istvan Mifsud, Jessica De Battista, the clinical consultants from the pathology, James De Gaetano, from surgery, Charles Cheney, and the navigators who took care of all the consenting, and of course the patients that consented to the study. And of course all this cannot happen without funds, therefore thank you to the grant holders, Malta Council for Science and Technology, and the RIDT Fund and Alive Foundation. And with that, I thank you, and I open the panel for any questions. Thank you very much.
Thank you, Dr. Grech, for that informative presentation. And we will now start the live Q&A portion of this webinar. If you have any questions you'd like to ask, please do so now. Just click on that Ask a Question box located on the far left of your screen. So let's get started. We already have some great questions coming in. Dr. Grech, an audience member says, very interesting. Can you explain what the added value of this method is compared to other PCR-based gene expression methods?
Okay, Helen, thank you. So here, the PCR-based methods, of course, will amplify the templates, meaning that the template has to be of adequate quality. And in our hands, when we compared PCR-based technologies with the quantigene technologies do better when the quality of the material is low. And Also, of course, if you look at multiplexing, then you can go up to 80 genes simultaneously with the quantity genes, whilst the gene expression profiles a method like real-time PCR that's limiting to the number of channels on the machine. And in addition, also, like, it's more sensitive, as I showed in the presentation. We can also go down to detecting 5 cells in a liquid biopsy setting. That means that it is extremely sensitive. We could not manage to do that with PCR methodologies. So I think the answer to this question is that it is advantageous for low quality material. It was also advantageous for low input material, and also for multiplexing up to 80 genes to now. And you of course, you can actually increase that as we go along with technology development.
Thank you so much. And can you clarify the CRC invasion panel and how this can be used in a patient setting?
Okay, so we started off with a panel of 40 gene amplifications in which we run various material, and we have identified specific areas in FFPE material and blocks, patient blocks, where we find a lot of heterogeneity, and therefore we have different sites. And one of these of particular interest is actually where we have the invasion front of the tumor itself. And here we identified specific gene amplifications. And also I've shown that gene amplifications actually also change in matter of between primary tumors and metastatic lesions. So it is very versatile, and the CRC invasion panel is very specific in that case because it's about the invasion front of the tumor. And that's one of the things that we are working on currently. In fact, we also put up a number of cellular models, which I did not present today, in which we have early response markers for invasion. And of course, these being part of panels like these would always be useful. And although during invasion we might not find circulating tumor cells within liquid biopsies, our hypothesis is that exosomes we can actually detect also early response genes during tumor development.
Thank you so much. And again, I want to thank our audience for these great questions coming in. We have time for a couple of more. This next question, for the gene amplifications in colorectal cancer, can one measure 40 genes in low input material, for example, microdissection material?
Okay, so the simple answer is yes, but I will explain as well. So we actually did a couple of experiments, and actually the first, when we put together this methodology, we also published a microdissection material paper in which we have very low input, and in that paper we also read 40 genes. So we had a panel of 40 genes, and we can actually read them. The interesting part is that we can use the quantity gene assay also on HE stained, so HE stained material from patients. So you take the slides, you HE stain them, and you actually can eventually also microdissect accurately on stained material and then go into the assay. Now, one of the advantages is, of course, you don't need to do RNA extraction, and you don't need to do cDNA extraction. And of course, you don't need to amplify the target, which means it's a very short workflow. And because of that short workflow, you maximize on the input material because you don't lose that much material as when we actually do different
methods to get to our endpoint measurement. So yes, we did work with microdissection material, but I would like to add the point that we can also use stained microdissection material so that you can select the regions more accurately.
Thank you so much. And it looks like we have one more question coming in, and this is a two-part question. Can you use this methodology for patient-derived xenograft models? And then the second part is, what are the advantages and proposed applications, if so?
Good. Yes, we did work in collaboration with other institutions in which we have used patient-derived xenografts. We actually set up whole tissue microarrays for the xenograft models, and the methodology can easily be used, of course, for this particular material. And the advantages are the same as I explained before. You can do multiplexing with various genes, and the workflow is very short, and the sensitivity is extremely high. So you can actually use also minimal input for these assays.
Dr. Grech, I want to thank you for your time and your presentation today. Would you like to provide any closing remarks before we go?
Yeah, so here, of course, it's a merge of technologies, and I would like to sort of mention that the use of signal amplification and bead-based assays in XMAP technology, which is a Luminex technology, is definitely something that we found to be very useful in terms of both time and workflow, because to do one assay, you really require an overnight hybridization step, and then the workflow is a 3-hour workflow. So in 3 hours, you can do 96 samples at one go. So with these technologies, we found ourselves very with high efficiency and efficacy in identifying also biomarkers and also validating biomarkers in such a very short workflow.
Dr. Grech, I want to thank you again for your time and for your important research. And before we go, I also want to thank our audience for joining us today and for their interesting questions. I'd also like to thank our sponsor, Luminex, for sponsoring today's webinar. Questions we did not have time for today and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. And this webcast can be viewed on demand for 2 years until September 2023. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Stay safe. Bye-bye.
Thank you.